We have very exciting talks ahead, but the Shanghai site seems to have a technical problem with the sound, so they have been fixing it. So Ling should come on the second position. So today's first presentation will be given by Denis LaFontaine from ULB, who will tell us about human diseases of ribosome biogenesis dysfunction.
Okay, so thank you. So let me first thank the organizers for a great meeting. It's always a pleasure for me to be back to Japan and Asia in general. I'm Denis LaFontaine. I'm from the University of Brussels in Belgium. And I'm here to talk about ribosomes and the disease we develop when we don't make our ribosome well. For many years, my lab has investigated ribosome biogenesis in eukaryotes, starting in budding yeast, and then we moved on to human cells, where we conducted multiple large-scale screenings, leading to the identification of several hundreds of transacting factors. At the same time, we developed an algorithm, a computer software, to characterize with a very high precision the nucleolar morphology, a technique which we call index of nucleolar disruption or INO scoring method, a method which received a patent in Japan 2 weeks ago. Using this method, we could address many different questions, one of which was to know which of the 80 ribosomal protein is important for nucleolar structure maintenance. And we found that out of these 80 ribosomal proteins, only very few were important for nucleolar structure. And in fact, it was mostly two, UL5 and UL18. And the reason why I'm naming these two proteins to you now, UL5 and UL18, is because, as we are going to discover together during my presentation, it seems that evolution has converged onto these two proteins to make them play very important roles even outside of the ribosome. Currently, my lab is investigating ribosome biogenesis dysfunction disease, and we do this using a range of animal models, and I'm showing you here a few examples of what we did over the last 2-3 years, using C. elegans, the fly, the fish, Xenopus, the mouse, and when it's available, we also like to use human-derived material, human cells. And that's what I'm going to talk to you about today. I'm going to pick on some of these examples, and I'm going to describe them to you. My aim really is to give you an overview of the kind of disease you can get when you don't make your ribosome well, and also highlight the multiplicity of models that one can use to address these diseases. The take-home of my lecture is going to be a very simple one, and that is that there is a need to produce a right amount of ribosome in cells in order to maintain homeostasis. If you make too much ribosome, it's not a good news because you're going to promote processes such as tumorigenesis, which may lead to cancer, and that's not something you want. And if you don't make enough, you're going to fall into a category of disease which we call ribosomopathies. Ribosomopathies are caused by an insufficient number of functional ribosomes in your cells. Ribosomopathies are often caused by mutation in ribosomal proteins or in ribosome assembly factors. And there's really something fascinating about this disease for us researchers, is that despite the fact that there are ribosomes in every single cell of your body, this disease will only impact particular tissues in your body. And the tissues which are primarily affected by this disease are the blood, leading to hematopoietic problems, and the brain, leading to neurodevelopmental disorders. So What I'm going to do today is I'm going to first start by the cancer perspective, and I'm going to tell you that it's possible to use ribosome biogenesis inhibitors, which are drugs, to kill preferentially cancer cells. And you can do that by activating a regulatory loop known as nucleolar surveillance. And I'm going to tell you about our latest finding in this field of nucleolar surveillance. And then I will move on to what happens when we don't make enough ribosomes. And I'll give you both examples of blood disease and brain diseases. So let me first remind you how complicated it is to make a ribosome in a eukaryotic cells. But let's say that I like to think about it with these 6 easy to remember keywords, which I listed here on the left. And it starts with the synthesis of the components which occurs in the nucleolus. And that's where RNA polymerase I synthesizes a long primary transcript, which sometimes we like to call polycistronic because it contains 3 of the 4 major ribosomal RNA. And this implies that this primary transcript has to be cleaved in order to produce the major ribosomal RNA, and these cleavages is what we refer to as processing step. There are many other steps that I'm not going to talk about: modification, packaging with the ribosomal protein, that's the assembly. There are many transport reactions, and also many quality control and surveillance reactions. But what I really want to tell you is that all of these steps are susceptible to go wrong. And so that means that they can be the subject of an inhibition. They can be targeted by an inhibition. And this implies that they can be targeted by a drug, which means that ribosome biogenesis is a highly druggable pathway, potentially. And indeed, at this stage, we have really good evidence that that's the case for the first step, the RNA synthesis step, for which we have drugs. The best known ones are listed here, CX5461 and BMH21. These 2 compounds actually target the first step, the ribosomal RNA synthesis step. They inhibit polymerase I, and in principle, they will kill any cells, but they preferentially kill cancer cells because cancer cells rely on heavy doses of ribosomes. They require more ribosome, and that makes them more sensitive to these compounds. And for this to happen, you have to activate a particular regulatory loop, which is p53 dependent loop, and which we call nucleolar surveillance. And that's what I'm going to explain to you now. But for that, I need to step back 1 step, and I need to show you a beautiful image of a ribosome with the small subunit in blue, 40S, involved in decoding the messenger RNA, shown in black. The large subunit here on top, the 60S, involved in amino acid polymerization. I'm going to crack open this ribosome, and I'm going to focus on the large subunit, on a structure which you have never heard about, the central protuberance. And what this structure is a trimeric complex which consists of 2 ribosomal proteins, UL5 and UL18, and 1 ribosomal RNA, the 5S RNA. So these 3 elements together, UL5, UL18, and 5S ribosomal RNA, they make a trimeric complex which is integrated into the large subunit, making the central protuberance. Now that you know what the central protuberance is, I can really explain what nucleolar surveillance is all about. In a normal cell that produces ribosome well, this trimeric complex is assembled during the assembly pathway into the maturing 60S subunit, and the cells are making their ribosomes and everything is good. And at the same time, p53, which is an anti-tumor protein, it's a good protein, but it's a protein which is going to kill dysfunctional cells, so you don't want to have too much around. And this is a normal cell. And indeed, p53 is constitutively degraded by ubiquitination by HDM2. So HDM2 is clicking a ubiquitin on p53, and p53 is degraded by the proteasome, so you don't have a lot of it, and the cell is happy and is making its ribosome. In a cell which is dysfunctional and cannot make its ribosome, such as in a cell which has been treated with a compound which inhibits RNA polymerase I, the trimeric complex is no longer incorporated into the maturing 60S subunit. Rather, this trimeric complex is going to accumulate freely in cells, and it's very stable, and it's quite abundant. And what it's going to do is that it's going to capture HDM2, and it's going to sequester HDM2 away from P53. Therefore, P53 is no longer going to be ubiquitinated, it's going to be stabilized, and P53 is a transcriptional factor which is going to activate a death program, and that's going to lead to the killing of the dysfunctional cell that can be the cancer cell. Now, I'm going to show you this with a very simple experiment, a Western blot. And in this lane, we have inhibited ribosome biogenesis by removing an assembly factor. The name doesn't really matter. And according to the principle that I've just explained, these cells accumulate a lot of P53. And to prove to you that this is really requiring this trimeric complex capturing HDM2, if you not only inhibit ribosome biogenesis by removing this assembly factor, but if you also remove UL18 or UL5, then you don't accumulate P53. And that's the principle of nucleolar surveillance, and it works really well. It's a very powerful mean to kill cancer cells. But the question was to know why is it that liquid cancers are responding very well to this regulatory loop, but the solid tumors were not responding as well to nucleolar surveillance? And that's something we didn't understand. And we started to address this question together with Tsuyoshi Osawa from the University of Tokyo. And together with Tsuyoshi, we reasoned that the reason why solid tumors are less responsive to ribosome biogenesis inhibitors is maybe due to the solid tumor microenvironment. And it was known that the solid tumor microenvironment is very heterogeneous in composition, and in particular, that at the core of the tumor, there are less nutrients. What we found was that indeed, there is less glutamine at the core of the tumor, and glutamine is very important for nucleolar surveillance activation. That's what I'm going to show you here with a simple Western blot again. These cells have been treated with CX5461 to inhibit ribosome biogenesis. And according to what we've just discussed, these cells accumulate a lot of P53. But if you remove glutamine from the medium, then suddenly this P53 accumulation doesn't occur anymore, which proves that glutamine concentration is very important for nucleolar surveillance activation. And this observation was highly specific because we could remove many other components from the medium, such as other amino acids, and we will not see this effect. So it was glutamine specific. We could also highlight precisely the concentration of glutamine, which is necessary to activate the regulatory loop and to lead to the stabilization of P53. We could show that this was not specific to only one ribosomal RNA synthesis inhibitor, but the same principle will be conserved with other drugs which also inhibit ribosomal RNA synthesis. We could also show that this works similarly well in many different cancer cell lines that we tested. And finally, we could start to delineate the signaling cascade which is involved in relaying the signal from glutamine to de novo synthesis of UL5 and UL18 and activation of the nucleolar surveillance loop. So glutamine is very important for nucleolar surveillance. And now I'm going to shift to what happens to when we don't make enough ribosome, and that's the ribosomopathy. And I will give you both cases of blood disease and brain disease. And I'll start with the blood disease, which is Diamond-Blackfan anemia. That's one of the best described ribosomopathy thus far. We've characterized a new protein, ETAR3, in this disease recently, and that's what I'm going to talk to you about. So Diamond-Blackfan anemia is a red blood cell disease, meaning that patients cannot make red blood cell, and they survive because of weekly blood transfusion, which is problematic because it causes a lot of iron overload in their blood. And that disease is caused by mutation in ribosomal protein, and to date, mutation in up to 24 different ribosomal proteins have been associated to this disease. But let's say that 25% of all patients worldwide have a mutation in a single ribosomal protein, RPS19. which is a protein from the small subunit head. We are part of 2 European consortiums that are actively monitoring this disease across Europe, trying to identify new genes and develop new treatments, new therapeutics, including gene replacement techniques. Within this consortium, we came across a new gene for which we identified 6 patients from 4 families of French, Turkish, and Dutch descent. All of these patients were harboring mutation in a protein called ETAR3, which is not a ribosomal protein. ETAR3 is not a ribosomal protein, but it has a yeast homologue called SIO1. And in yeast, it had been shown that SIO1 is important for the nuclear import of a ribosomal protein called UL18. So SIO1 will pick UL18 in the cytoplasm as it is being synthesized by the ribosome and will drive UL18 into the nucleus. So considering this, the first thing we wanted to do was to test if this is also the case for the human protein ETAR3. And we performed many experiments to test that, but I'm showing you here an immunostaining on a model cell line ELA cells, for which we depleted ETAR3 by use of silencers. In a control cell, UL18 was detected at 2 places in the cells, in the cytoplasm, where ribosomes are engaged in translation, and in the nucleolus, where ribosomes are assembled. In the cells depleted of ETAR3, we lose the signal in the nucleus and in the nucleolus, and that confirmed that ETAR3 in human cells is playing the same role as SIO1 in yeast. That is, it's a ribosomal protein transporter. For us, it was very important because it's one of the first non-ribosomal protein gene associated with the disease. Nonetheless, it's a ribosomal protein transporter, so it makes sense. We could confirm these results by using human cells directly from the patient. The patient cells also had less UL18 in their nucleus. We could show that the patients are defective for ribosomal RNA processing, and that processing defect could be complemented by an antiviral construct expressing the wild-type gene. And using CD34 plus cells purified from the patient, we could show that these cells cannot differentiate into erythroid. Now, in the few remaining minutes of my talk, I'm going to talk about the brain diseases, and I will mention 2 cases. The first one is the Labrune syndrome, which is a very bad disease in which patients accumulate cysts in their brain. And the disease is also called lonkoencephalopathy with cerebral calcification and cyst. It's a very interesting disease because it's a monogenetic disease which is caused by mutation in a single gene, U8. U8, small nucleolar RNA, which is a SNO RNA involved in the removal of the 3' external transcribe spacer of the primary transcript that we mentioned at the beginning of the presentation. If you use model cells and if you remove U8 with azo silencers, and if you perform a Northern blot with a probe that is specific to the 3' ETS, in a control cell, you only detect the primary transcript, but in a cell without U8, you start to detect all sorts of precursors which have retained the 3' ETS onto them. If you analyze patients suffering from Labrune syndrome, the first thing you see is that they have less U8 at steady state. And if you perform the same Northern blot analysis, you realize that these patients also accumulate strongly these 3' ETS, these species which have retained the 3' ETS sequence onto them. For this disease, we could model the disease in a fish, and the fish showed many of the phenotypes of the human patient, including trunk vasculature problem. I'm now closing my talk with 2 methyltransferases, BUT23 and METL5, which are 2 methyltransferases acting on the small ribosomal subunit. BUT23 is adding an M7G methylation mark in the head of the small subunit, and METL5 is adding an M6A methylation site mark right at the decoding site. These 2 methylation marks are actually facing each other across the mRNA channel. The mRNA is actually wrapping around the small subunit, around the neck like a scarf, and these 2 modifications are actually facing each other around this channel. We've been working on these modification enzymes for many years, but recently we learned of patient mutation in these methyltransferases, and these patients actually suffer from rather similar clinical manifestation, but which can be summarized by intellectual disability. They have specificities, but they share many of their symptoms, one of which is unbalanced walking. I'm going to focus on the unbalanced walking phenotype and on MEDAL5 for the sake of time. And because we could produce a fly model with a mutation in MEDAL5. And the fly is a very powerful model because it has a lot of behavioral assays available, including one assay called the Buridan paradigm, on which you put your fly on a disc. You put your disc on a little water dish. This is closed in an arena, and you can actually follow the walking behavior of a fly. A normal fly will walk from one black stripe to the other pretty much in straight lines, which you can record with a video camera. But with the MEDAL5 mutant fly, we could observe a very disorientated walking behavior, which was very reminiscent to the phenotype of the actual patient suffering from this neurological disorder. So I hope that I could convince you that there is a need to produce a right amount of ribosomes in your cell, and if you make too much, you might lead to processes such as tumorigenesis. If you don't make enough, you're going to develop highly tissue-specific diseases, and we've mentioned blood and brain. And I hope that I also convince you that it's possible to kill cancer cells by triggering a nucleolar surveillance pathway, which requires p53. But for this to happen, there is a need to know about the microtumor environment, and in our case, we have pinpointed the importance of glutamine in this activation. So I'd like to thank the people who were involved in this work, and in particular, Christian Zorbas, who was involved in most of these experiments that I mentioned, and my technician Ludivine Wachel, with contribution from other members of the team. We are recruiting 2 postdocs in the lab currently. I also would like to thank very much my many and great collaborators worldwide, including Tsuyoshi Osawa and Tsutomu Suzuki at the University of Tokyo, with whom we developed the glutamine work, Jean-Yves Roignan in Lausanne, with whom we developed the fly, the MEDAL5 fly. Mark Hai and I have been characterizing these methyltransferases together for many years. Yannick Rowe was involved in the Labrun syndrome disease. And now I would like to acknowledge my funding as well, with a particular mention for the European Joint Program on Rare Disease, which is financing the research on Diamond-Blackfan anemia. I think my time is up. Do I still have time? I still have 1 minute. Then I will like to flash quickly a work we did recently with Damien Hermand in Namur, where we characterized an entirely new type of RNA modification on the messenger RNA, dehydrouridine, and we showed that dehydrouridine is actually slowing the ribosome, and that's why we used this little symbol of a snail to embody the idea that when the ribosome encounters dehydrouridine on messenger RNA, is actually slowed down quite a bit, and we showed that this is important for meiosis in fish and yeast, and the work was actually conducted in my own town, Namur, and the symbol of Namur is actually a snail, so it was quite nice for us to use this. And I also would like to tell, for those who are interested by ribosomal RNA modification and RNA modification in general, that we have re-established a complete pipeline of direct RNA sequencing by nanopore to remap quantitatively all ribosomal RNA modification using both the MinION and the smaller fungal devices. In case you are interested, you can come to me later on, and we'll be very happy to discuss about this together with you. Thank you very much.
Thank you. Do we have questions from Shanghai side?
Yes. Hi, a very last talk. So my question is, how the nuclear surveillance pathway is specifically activated by the glutamine, but not other amino acid? Is there any relationship with the mTOR pathway?
Excellent question. So I can't say that we've cracked everything, but what I can tell is that clearly we depleted the other amino acids, and when we remove the other amino acids, we don't see this effect that glutamine has to suppress the activation of nucleolar surveillance activation. About the TOR pathway, glutamine is very well known to activate the TOR pathway, and indeed we showed that the TOR pathway is activated in this context. We could see that by phosphorylation, increased phosphorylation of protein S6. Nonetheless, we could also use taurine and rapamycin, which inhibit the TOR pathway, and this actually didn't have any impact on nucleolar surveillance. So although the TOR pathway is activated by glutamine, it's not directly involved in nucleolar surveillance activation.
Okay.
Now, how glutamine triggers a cascade that leads to de novo synthesis of UL5 and UL18, what we have shown by use of a combination of inhibitors was that it involved the RAS-RAF-MEK-ERK pathway leading to the inhibition of EIF2K, and that promotes global protein synthesis by EIF2. That's what I can tell. That's how much we know for the moment.
Okay, so is there any structural information about the HART3 protein? Structural information?
Alors, we don't have information about the human protein ETAR3, but we have information about the SIO1 protein, which was performed by Roland Beckman in Munich. And that protein actually it's very well characterized how it interacts with ribosomal protein, but we don't have the equivalent structural information for the ETAR3. But of course, ETAR3 and SIO1 are quite well conserved. So in principle, we could base our rationale on the structural information we have for the yeast complex.
Okay, thank you.
You're welcome.
Hi, Dennis. Very nice talk. I want to ask one question regarding with the p53 and the inhibitor. Is the inhibitor specifically like upregulate the p53 or other proteins also get affected?
So the nucleolar surveillance pathway, which is p53 dependent, is only part of the picture, basically. And it's, of course, it's a simplification and it's a bit of a caricature of a situation. There are more important proteins such as retinoblastoma and other disease proteins, which are also activated by ribosomal proteins. So ribosomal proteins are very abundant. And I think during evolution, they have been used for other functions outside of the ribosome. So the case of UL5, UL18, and 5S, together with p53, is one very important regulatory loop, but certainly there are more loops to be identified, maybe on the same or little variation of that principle.
Thank you.
That's the best known so far.
So maybe you have been asked the same question. Why this kind of ribosomopathy affect only a certain tissue or cell types? Or because the ribosome is required for every cell, but always this kind of
that's a very good question. We would love to know the answer for, but we don't. But we have ideas. So the idea is that probably cells are making too much ribosomes, right? I mean, let's say they are making 100 ribosomes, but let's say a skin cell is happy with 50 ribosomes. Okay, it's making 100, but if it were making 50, it will still be fine. But then if you are a neuronal cells or maybe a blood precursor cell, you're not fine. You need the full complement. You need your 100, and the 50 is not gonna cut it. So that's the idea. So the idea is that some cells require a full complement of ribosomes, and some cells can do well even if they don't have the full production of ribosomes. And what we observe is that it's often during cell differentiation pathways or at particular stages during embryogenesis that there is a particular need for these larger, let's say, amounts of ribosome.
Or are there any possibility that the heterogeneity of the ribosomes is affected by just...
Yes. So there are currently two models which are coexisting, and which I like to think about two coexisting models rather than two opposing models. On the one hand, there is this quantity type of model where people think about the problem in terms of how much ribosome is produced. That's why I use the term number of functional ribosomes. And on the other, there is this view that says that cells are producing different types of ribosomes. And these ribosomes can differ by protein composition, but mostly by ribosomal RNA modification composition. And we ourselves in my lab have shown that there are indeed ribosomes carrying more or less modification. So we know that's reality. I mean, we know that in cells, there is not a single type of ribosomes. There are many different types of ribosomes, blue ribosome, green ribosomes, red ribosomes. And these ribosomes are actually characterized by a different quantity of ribosomal RNA modification, which suggests that they may translate differentially, but doesn't prove that they translate differentially all the time. However, there are cases where it has been shown quite nicely that differentially modified ribosomes do translate differentially. So I think there's scope for coexistence of both models. On one side, the amount of ribosomes. And of course, if you reduce the amount of ribosomes, the messenger RNAs, which are naturally less translated, will even be less translated because you further reduce the overall amount of ribosomes. And then there is this model of ribosome specialization or ribosome heterogeneity, where a green ribosome translates a green messenger RNA. And I think this has only been proven well in few cases, but I think there's certainly room for still thinking about that model together with the quantity type of model.
Just the last question. Do you think that nucleolar surveillance affects the content of the ribosomal proteins of the, isotype of the ribosomal content?
I didn't catch the first part of the question. If the
nuclear surveillance is activated.
Oh, the surveillance.
Right. Just a total downregulation, or this may change the expression ratio of the isotypes of ribosomal.
Yes, I mean, the term surveillance covers many different aspects. So, the nucleolar surveillance is a regulatory loop which really kills dysfunctional cells. That's one type of surveillance. There's quality proof control, which we also call surveillance. And Angus Lehman, 20 years ago, has showed that cells by default are producing far more ribosome than they need. They produce an excess of ribosome, and these ribosomes are turned over very rapidly. So you don't see them unless you use proteasomal inhibitor, then you see them coming out. So there is a massive and constitutive degradation of excess production of ribosomes, and that allows cells to react, to respond very quickly in case there's a need for more abundant production of ribosomes. So your question is about whether that kind of surveillance / degradation might influence the activation of the nucleolar surveillance, the p53 loop. Yes, it might, in a way that if it leads to more or less accumulation of this trimeric complex that I described, which is catching HDM2. So that's really the key. I mean, it's a very stable complex, stable outside of the ribosome, and it's going to sequester and catch HDM2. So if you activate degradation of ribosome leading to accumulation of this trimeric complex, then yes.
Thank you so much.
So I'm very much interested in the p53 activation is affected by the external glutamine concentration. That it's also affected by the changes of the metabolic pathway that affect the internal concentration of glutamine.
So I don't have a lot of information about this right now. A lot of the work we've done was performing culture cells, but we have still performed a fair amount of work in cancer xenograft in mouse. And we have extracted tumors from mouse xenograft, and we have analyzed pre-ribosomal RNA processing and glutamine and nutrient concentration in the core fraction of these xenograft and in the, let's say, peripheral area of the tumors, which correlated very well with the cell line work. Yeah, so that's what I can tell, but I don't have more information about that.
Thank you very much.
And we haven't investigated how metabolism fueling into glutamine might be important or not for the overall process. This is something that is open for the future.
Thank you very much.
Thank you for the great presentation.